My name is Diane O'Dowd, and I'm a professor in the departments of Anatomy and Neurobiology and Developmental and Cell Biology here at UC Irvine. Today I'm going to tell you about the preparation of neuronal cultures from Drosophila pupae. This involves removing the brain from late-stage pupae, and we generally culture just the central brain region, and at this stage, the major anatomical structures that are present in the adult brain are already formed. This includes the antennal lobes, where there are projection neurons that send their axons all the way up to the mushroom body region, and the mushroom body neurons, Kenyan cells, which have sent their axons down to their target neurons through the alpha, beta, and gamma lobes. On the day of culturing, the first thing we do is go into the laminar flow hood and we prepare sterilely the enzyme solution that we're going to use to dissociate the tissue. And we also make up the medium that we're going to use for growing the cells, and this is made from stalks that we've prepared ahead of time. The next step is to collect the pupae that we're going to use for this for the dissection, and that's done outside of the hood because these are grown in vials that contain yeast and all sorts of other things, and so we actually do the initial stages of our dissection outside of the hood. These are pupae that